Yeah. Hello, Pawan. Hello.
Hello. Hello.
Hello.
Brahmaya.
Good afternoon, Pawan sir. Good afternoon.
Good afternoon.
Good afternoon, Mohan Verma sir. Good afternoon.
Hello, sir.
So good afternoon. We are all hello, sir Mohan Verma.
Yes, you are audible.
Sir,
You're
good
audible.
afternoon, Mohan Verma, sir. Good afternoon, sir. Good afternoon, Brahmaya. Good afternoon, Natras, sir.
Good afternoon, dear all participants. Thank you all. Today we are having I Pawan Kumar sir, senior scientist, the Alphabet Foundation Private Limited. So Mohan Verma. Mohan Verma. Over to ఇక్కడ మాట్లాడేసింది. Good afternoon all. So today we are having Today we are having our renowned alumni, I. Pawan Kumar. So, sir is working as a senior scientist in the AlphaMed Formulation Private Limited. And then requesting all for FDP3, that is day 3 program, that is 10/1/24. So, over to Mohan Verma, sir.
Dear staff and students, I feel very happy. We are having a very eminent alumni from SUCP. He will be addressing in this FDP. He will explain about the recent trends and the innovations in the industry. He is highly experienced in the area of the FR&D. So the students and staff, if you have any doubts about the industry, you can just interact with him. So I feel very happy. And I wish you all the best. Thank you.
Thank you, Mohan Verma, sir. Thank you so much. And then, so now, Madam is there. Divya Madam is there. Madam will give introduction regarding Pawan Kumar, sir. So, requesting Madam to give the introduction regarding Pawan Kumar, sir.
A very good afternoon to everyone. I'm Divya, Assistant Professor from Shri Vishnu College of Pharmacy. And today I take this opportunity to introduce our today's speaker, Mr. Pawan Kumar Inuganti. Mr. Pawan Kumar Inuganti is a pharmaceutical industry professional with more than 10 years of experience in formulation, research and development of solid orals, liquids and powder from oral solutions. So he took his B.Pharm degree from Vagdevi College of Pharmacy and M.Pharm degree from Shri Vishnu College of Pharmacy. Coming to his working experience, Pawan sir started his career in 2011 as a project trainee at Aurobindo Pharma Limited, where he worked for 6 months. And then he worked as an executive at Dr. Reddy's Laboratories for the span of 11 months. And then at Medreach Limited, he worked as an office research and development for a span of 1 year, 3 months. And he worked as a senior research associate at AlphaMed Formulations for a span of 1 year, 6 months. And then he worked for a span of 6 years, 11 months in Aurobindo Pharma. And in Aurobindo Pharma Limited, he worked as a research associate 3, research associate 4, scientist 1, and scientist 2. And presently, sir is working at Aspen Pharma Group as senior scientist 1. And with this brief introduction, I hand over today's session to our beloved speaker, Pawan Kumar, sir. Thank you.
Good afternoon, all. Good afternoon, Mohan Verma, sir.
Good afternoon, good afternoon. Good afternoon.
And good afternoon, ladies and gentlemen. I stand before you today with great enthusiasm and gratitude as we gather for 1 week online national FTP SDP seminar focusing on trends in pharmaceutical industry. Okay. Just I give you a brief introduction on overview of technology transfer of pharmaceutical products, because already you have seen what is the product development and all. I'll summarize some of the things from industry. Over to the next slide, please. Next slide. 1 second, I'll share the screen.
Sir, is visible?
Yeah, it is visible. 1 second, I'll share the screen. Is my screen visible to all?
Yeah.
Hello?
Yeah. It is visible. Visible, Pawan sir, is it visible?
Okay, first of all, I Start with introduction. According to World Health Organization, technology transfer is a logical process that controls transfer of any process together with its documentation, professional expertise between development and manufacture or between the manufacturing sites. Okay, it is a very logical procedure, and there will be a technology transfer between R&D and manufacturing site, and the other one is between two manufacturing sites. Because if there will be unit 1, unit 2 in same company, there will be transfer from one unit to other unit or from R&D to the unit. Okay. Second point, it is a systematic process that is followed in order to pass the documented knowledge and experience gained during development and or commercialization to an appropriate, responsible, and authorized party. So it is both the things will be uh, the documentation part and the experience gained during development part. Both should be incorporated into the technology transfer process. Otherwise, the process will get collapsed. Okay. Third point, technology transfer embodies both the transfer of documentation and the demonstrated ability of receiving unit. The one which is sending the documents means R&D will be the sending unit, and the one which is receiving manufacturing site will be the receiving unit. Okay. Here, to effectively perform the transfer technology to the satisfaction of all the parties and regulatory bodies, both the documentation part and the technology part should be parallelly transferred to the site. Okay. Overview of technology transfer in pharmaceutical manufacturing. In this, we start with phase 1, that is project initiation. Transfer from development to commercial site, or a transfer from one company to another company or another unit. Previously, what we have discussed is already it is between R&D and commercial plant, or in between two different plants. Okay, the receiving unit, the one which is receiving the dossier will be treated as receiving unit. The one which is sending the dossier will be treated as sending unit. It should be able to accommodate the facilities and intended activities should be monitored. Okay, the receiving unit should have technical expertise, technology, and capability to transform the product from one side to another side. Second one is project planning. This planning includes sending unit and receiving unit should jointly establish a team. They both should coordinate the activities simultaneously, and if there is any gap between the two units or between R&D and plant, they should be addressed immediately. And technology transfer document should be maintained, and technology transfer master plan along with protocol should be prepared. Specifications and critical material attributes should from R&D receiving unit and sending unit should be same. The sending unit and receiving unit information with respect to qualitative and quantitative composition, physical description, manufacturing methods, controls, specific and packing components, configuration, safety and handling consideration should be keep in handy. Otherwise, any of the process has been collapsed, then the product will be damaged and we cannot send it to the market. A detailed description of facility requirements and equipments. Suppose if product is moisture sensitive, so the uh, requirements and facility able to produce RH less than 30, 20, a specific requirement should be keep in mind. Project transfer execution. After analyzing the gap between the sending unit and receiving unit, the transfer execution should identify any difference in facilities, systems, and capabilities, and these should be discussed with sending unit. The sending unit should cooperate with receiving unit and understand potential impact and satisfactorily address to assure the equivalent and bioequivalent product so that patient compliance will be addressed. Based on the information received from sending unit, the receiving unit should consider its own capability to manufacture and pack the product to the required standards. and there should not be any market complaints or customer complaints should be received, because many of the technology transfer related problems nowadays we are facing is market complaints and customer complaints. These complaints may first maybe major, minor, and moderate. If the major complaint is received for a particular product for more than 3 to 4 times, the product will be audited by the regulatory agency. So it should be handled properly. The third process already is project execution and continuation of project execution, comparison and assessment of suitability and qualification of facility and equipment, manufacturing process, flow, personal and critical steps, including hold time studies, endpoint of granulation, sampling techniques, sampling points, and compliance with regulatory requirements, or any changes which are required should be evaluated. The fourth phase will be a review of all the documents and closeout. Then we come to product development. What are the components of drug product? Drug substance, excipients, formulation development, warrages, physicochemical and biological properties of drug product, manufacturing process development of drug product, container closure system, microbiological attributes, and compatibility of drug substance with excipient. What are the elements of product development? Quality target product profile. Already, I think all of aware of this QTPP CQA parameters, everything, and we have to arrive design space. Life cycle of the product should be maintained. If a particular product is developed and stability is established, and if we are not able to generate that product for 10 to 15 commercial batches, then that will be compromised. So the proven acceptable range should be kept in mind. Overall pharmaceutical development includes manufacturing process, process controls, product specification, control strategy, and life cycle management. These should be properly evaluated before starting any project. And coming to the overall product development steps, when we will get a project brief or project code, and we'll start licensing activities, evaluation of API. After evaluation of API, we will take one small scale batch and it will be submitted for analytical method development. After the different product characterization, selection of excipients. If any of the excipient we are facing any issue with respect to quality targeted product profile, then the excipient will be changed. Packing systems, compatibility studies, prototype development, and stability studies. Pre-screening stability studies will be done at lab scale batches, and comparative dissolution testing with respect to a reference product. Already all you are aware of this F2 matching and all. I think hope you are everyone is aware of these things. And if there is any preservative in formulation, preservative efficacy testings, screening stabilities, and process optimization studies, finalization of specifications, method validations. Once method is validated, your formula will be freezed. If your formula is freezed, and if your method is validated, you cannot change any quantitative or qualitative composition of the formula. If you are changing anything, then your method validation should be redone. Otherwise, it should be partial. Maybe based on the changes you are doing, it will become partial to major validation, full-fledged validation. So pre-scale up activities, scale up batches, if required, pilot equivalent studies, stability studies of scale up batches, tech transfer package. So these are the just steps what have brief for your understanding and uh, uh, information purpose only. What is next? Coming to the next slide. What is product life cycle? What is product life cycle? R&D development, process optimization studies, scale-up batch, pre-exhibit batch, if required, exhibit batches. After finalization of formulation at R&D, we should prepare one master product document. The master product document will be having each and every step, how you are doing that formula into your finished product, starting from dispensing to final coating or a finished product. What is maybe if it is coating, then up to coating. If it is capsule filling, up to capsule filling. The manufacturing product document will be again in the same way. Manufacturing packing document will also be there. Manufacturing product document and packing document. Packing people will prepare packing document, and the packing configurations and packing structures will be finalized with respect to packaging development. And final tool drawing, raw material specifications. What are the raw materials we are using? That those specifications will be freezed, and those specifications should be shared to the sending unit, whatever manufacturing unit we have identified. Drug substance specification and STP, drug product specifications and STP. In process specifications, general tests such as blend uniformity, blend assay, split off studies, and cleaning methods. Last and finally, submission of formulation technical details of drug product. Coming to stage 2, process optimization studies. What are process optimization studies? Once your formula is freezed, your formula shall be capable of reproducing the same results within the specification limits what you have predefined. So you have to share bill of materials, BOM, to the plant and exhibit batches based on your requirement. Presentation of development data, including your stability, XRD, polymorphism, photostability, etc., will be shared to the plant. Then the plant will summarize with scale-up BMRs and scale-up matrix and protocols preparation. Process optimization batches execution shall be done as per pre-planned matrix. So your process optimization batches will be such as high fluid uptake, low fluid uptake, high hardness high hardness challenges, and speed challenges will be taken. R&D and process optimization data after presentation, final MPD shall be prepared for the commercial batches manufacturing or exhibit batches manufacturing. Here, data gathering should be done. Next, coming to stage 4, exhibit batches. After process optimization batches, if required, pre-EB shall be done with one lot of EB scale batch or same occupancy as EB scale batch. Otherwise, we can go directly to exhibit batches. If control release formulation, we prefer to go for pre-EB batches. At EB stage, we master product document, packing plan, drug substance, raw material specifications, everything will be finalized, and our formula will be freezed. With respect to analytical validation will be done, and analytical method transfer to the plant also will be taken place. After that, stability protocol as per our guidelines and as per the targeted market will be prepared. Along with all these technology transfer documents, including API specification, formula composition, forages, will be finalized and submitted. This is for intra-site products, because whatever we manufactured in R&D and we are scaling up to the site. And for inter-site, if it is between two different sites, and if it manufacturing site is from one unit to other unit, the technology transfer document should include API, active pharmaceutical ingredient or drug substance, excipients, formula composition, forages if any, manufacturing related documents and dossier sections, analytical related documents and dossier sections, packing related information, product history, if the any market complaints or any customer related complaints, PQR, product quality report, deviations, change controls, recent changes, and relevant dossier sections shall be submitted. Cleaning procedures most important, because if cleaning is not done properly, carryover chances may happen, and gap should be assessed between the two different sites. Technical feasibility assessment. After getting the project, it will be evaluated technically for feasibility. It is evaluated for manufacturing feasibility assessment, batch size proposals, analytical feasibility. These batches will be freezed based on our annual volumes of the particular product identified for the particular market. If our annual volumes are less, then the batches will be commercial batches will be less. Our annual volumes are more, our commercial batches will be more. So based on our annual volume, these batches freezing will be done. The regulatory impact of the drug product in our country or with respect to unit shall be evaluated. Licensing activities and cleaning evaluation STPs, related STPs shall be taken in place. Lab scale feasibility evaluation, scale up batch documents, technology transfer protocols, critical quality attributes, critical process parameters, and all manufacturing related activities shall be evaluated for the feasibility at the new site. The technology transfer report shall be prepared. Analytical method transfer along with process validation batches shall be taken place. With respect to inter-site products, formulation team will take a lab scale feasibility trial and they evaluate all the manufacturing process with respect to lab scale feasibility evaluation report. Technology transfer team manufacture feasibility assessment report, protocols and certificates will be done. Analytical team will prepare analytical feasibility assessment reports. QA team coordination of all activities. QC team will ensure the analytical method transfer. Engineering team will ensure the facility related activities. Packing will be responsible for packing related activities. After if everything feasibility assessment has happened, the new product will be introduced into the new manufacturing site with all proper controls and BMR, BPR, batch manufacturing records, batch processing records, process validation protocols, whole time study, stability study, finished product specification, standard testing procedures, raw material and material specifications will be prepared at new site. Analytical team will prepare in-house CYs for new materials and vendor qualifications for new equipments will be prepared. Based on our batch sizes, bill of materials and procurement of raw materials shall be done. Process validation will be done on 3 batches along with stability studies. After successful technology transfer, a technology transfer certificate will be provided to the sending team. Thank you.
Thank you, Pawan sir. Thank you for valuable information. So, Pawan sir has discussed regarding R&D development that are taking industry, and then what are the technology transfer that is going on industry, and then what are different process optimization studies has to be done during the formulation of a dosage from etc. etc. And then what are the inter-site products required and what are the GMP requirement and so Pawan sir given a broad explanation regarding different technologies, different processes that are happened in the pharmaceutical industry. Thank you so much, Pawan sir, for your valuable cooperation and valuable time and joining in the meeting. And then, so this is now questionnaire section. So any questions are there? Can you ask the Pawan sir? Sir? Any doubts, please ask Pawan sir. Dear participants, any doubts are there, you can ask Pawan sir. Sir, you explained well, that's why nobody is asking any questions.
Thank you. I extend my heartfelt gratitude to Vishnu College of Pharmacy, Dr. Mohan Verma sir, Dr. Natraj sir, Swami sir, and Brahmaya sir, and entire college students and students who have participated in this seminar. It was truly an honor to share my insights and perspectives with all these bright and uh, brilliant minds. The experience, what I have gained is very less, and whatever theoretical knowledge we are gaining from the institutions and colleges is more than the practical knowledge we are sharing here. And there is lot of things need to be shared. Just this is an brief and overview of an technology transfer and pharmaceutical development. Whenever a development had happened, that product or that formula should be feasible, and it should be production friendly, manufacturing friendly. If it is manufacturing friendly, then we can commercialize that formula into any number of dosage forms. If it is not manufacturing friendly, then we cannot manufacture at commercial level. So we adopt many techniques in bringing the product from R&D level to shop floor with maximum adaptability and feasibility. So, and I lastly thank you all for privilege and look forward for future opportunities for collaboration in such activities in person also. Thank you all.
Thank you, Pawan. Thank you so much. Thank you for joining in the meeting, and thank you for your valuable cooperation with our participants. And then you have given a wide information and enriched our participants with our knowledge. Thank you so much, Pawan sir. So thank you participants for joining in the meeting. Thank you all. Hereby I am ending the meeting. Thank you, Pawan sir. Thank you so much.
Thank you. Bye bye. All the best to all. Thank you, sir. Mohan Verma sir. Thank you.